Yeah, thank you for the introduction. Just to make sure before starting, can you see my slides?
Yes, everything is in order.
Okay, great. Thank you. So thank you so much for the introduction. I really want to thank the organizers for the opportunity to participate in this wonderful symposium. Congratulations on this very nice event. So today I'd like to talk about applications of chemoinformatics in drug discovery, and then present a personal view of challenges that is facing chemoinformatics and computer-aided drug design in general. So this is the outline of the talk. First, I'd like to present a brief introduction of our university, and just for the sake of formalism, just to state what chemoinformatics is, then we will move to a case study that is focused on applications of chemoinformatics in epigenetic drug discovery. In the next section, I will present this personal perspective of the challenges of chemoinformatics, and at the end is presented a brief summary. So the National Autonomous University of Mexico, or UNAM, where I'm actually right now, it's a nationwide university with around 350,000 students. The main campus is located in Mexico City in the south part. You're very welcome to visit whenever you have the chance. There are examples here of some of the buildings that are in the main campus. And of course, we have a soccer stadium. UNAM has extension schools in the US and also in other countries. This is our research group that is called DIFACHIM. DIFACHIM is an acronym in Spanish that stands for Computer-Aided Drug Design at the School of Chemistry. Today, and as always, I'm talking on behalf of all of them that are actually making the contributions. And just today, for instance, we listen to the talks of Ana Chavez and Edgar Lopez. So again, just for the sake of formalism, in this slide, we're showing one of the several definitions of chemoinformatics that has been published in the literature. And as everybody in this audience are, I'm sure, familiar with the main differences between these two main disciplines, bioinformatics, chemoinformatics. Bioinformatics, of course, is more focused on larger molecular systems. Chemoinformatics is focused on small molecules, and to this end, bioinformatics typically uses representations like sequences, 3D coordinates, while chemoinformatics uses molecular fingerprints, numerical descriptors, and others. Now, both disciplines and several other informatics-related disciplines have been around since the late 50s. Perhaps one of the most well-established disciplines by itself is bioinformatics. At least the name of chemoinformatics, as you know, was published in the literature until 1998. But in any case, all these informatics disciplines now have several applications, and in this symposium, we have seen very nice examples. For instance, toxicity prediction, applications of natural product research, biomedical informatics, and several others. A couple of more that I would like to emphasize here is epigenetic drug discovery. That, well, this name has appeared in the literature, epi-informatics, and also applications of informatics in food science or food informatics. And then in the next slides, I'm going to focus on some applications of chemoinformatics in epigenetic drug discovery. So now we're going to move to the next section, presenting this case study. And for this part, also give a brief introduction on epigenetics. Epigenetics are modifications that happen to the chromosomes without alteration of the DNA sequence that leads to an unstable phenotype. There are three major epigenetic protein families: writers, erasers, and readers. We know that histone methylation and acetylation are frequently dysregulated in cancer cells and other diseases. Methylation perhaps was the first epigenetic modification that was linked to cancer. And as we will see in the next slide, this process of methylation is regulated by a family of enzymes, the DNA methyltransferases. So over the last few years, we have seen an increase in the number of peer-reviewed publications linking epigenetics with several diseases. As I mentioned, the most well-studied so far, historically speaking, has been cancer, but in the last few years, we've seen more and more publications linking epigenetics with other diseases like diabetes, neurodegenerative diseases, cardiovascular diseases, and others. Also, here in the bottom part of the slide, we can see a clearly increase in the number of peer-reviewed publications that are investigating approaches for epigenetic drug discovery. And more recently, computational methods of various kinds have been applied to support these efforts. Here we're showing small molecules that are already approved by the FDA for the treatment of cancer. These small molecules hit different epigenetic targets, as stated here. Here in the bottom part of the slide are shown examples of other small molecules that are under clinical development. There are around 30 or 40 small molecules overall that are in different phases of clinical stages, and these target either epigenetic writers, erasers, and readers. And this review here at the bottom of the slide presents a really comprehensive overview of the current status of epigenetic drug discovery. So as I said, DNA methylation is one of these epigenetic modifications, and specifically DNA methylation is the addition of a methyl group to the carbon number 5 of cytosine residues. This process, mediated by this family of DNA methyltransferases, or DNMTs. We know that in cancer cells, overall there is an overexpression of these DNMTs, and also there are publications demonstrating the hypermethylation of tumor suppressor genes. There are two DNA methyltransferases that are de novo, DNA methyltransferase A and B, and there is one other DNMT, DNMT1, that is associated with the maintenance of these epigenetic marks. All of the DNMTs have a regulatory domain, and most of them, except 3L, have a catalytic domain. In the literature, in particular in the public domain, in Protein Data Bank, we can see that there are now many crystallographic structures of DNMTs. In this table are shown just few examples. Here I would like to highlight this paper by GlaxoSmithKline that in September of 2020, last year, published the first crystallographic structure of human DNMT1 bound to a small molecule. This compound was identified from a high-throughput screening campaign with around 6 million molecules tested, and the results show that this compound is a really potent DNMT1 inhibitor with an IC50 of 36 nanomolar, and is also quite selective. Did not inhibit 3A and 3B. This slide shows examples of small molecules that are known to inhibit DNA methyltransferases or the methylate cytosine residues in a cellular context. Among these several small molecules, the couple of here are cytosine analogs, 5-azacitidine and 5-azadeoxycytidine. These compounds are extremely potent molecules and are actually approved by the FDA for the treatment of myelodysplastic syndrome. However, because of the chemical nature, because of the chemical scaffold of these compounds are also quite cytotoxic. So for this reason, the scientific community has been searching for other small molecules that are also potent, effective, but that they have a different scaffold. Examples of natural products are these two. There are several other natural products available in reviews. There are also efforts to repurpose drugs now for the treatment of cancer, hitting DNA methyltransferases. There are also several examples in the literature where virtual screening, either ligand-based or structure-based, has led to the identification of promising inhibitors. Also, medicinal chemistry efforts doing synthesis have also identified promising compounds, and high-throughput screening has also put forward some small molecules. The most recent example is this compound from GlaxoSmithKline that I just mentioned a few slides before. So with this very brief background, now I can say that the goal of our research program in a group is the discovery and development of inhibitors of DNMTs and other epigenetic targets. The main approach to reach this goal is combine computational methods with the corresponding experimental validation. And in the next slides, I'm going to focus on two specific aims. One is the development of predictive models using machine learning, and the second one is the application of those models and other approaches to do virtual screening of compound libraries and identify new candidates. Now let's start with the first specific aim. And well, also everybody in this audience know that large databases like PubChem or ChemBL are extremely rich resources for structure activity relationships with millions of data points. So this was our starting point. And the first step here was to extract the SAR of small molecules reported with activity with 55 epigenetic targets. This table here, this study was performed by Jesus Naveja. The table here shows part of that information, a brief summary indicating the number of small molecules that are contained specifically here in Campbell. For instance, for DNMT1, 248 molecules. For DNMT1, this is the number of different scaffolds. This is the Bemis and Murco scaffolds and the percentage of active molecules, considering a threshold or 10 micromolar. And in that basically survey of Campbell, notice that there are other epigenetic targets that has been heavily explored, like HDACs. For instance, for HDAC1, at least in Campbell, there are more than 3,000 compounds. Anyway, so all the SAR data in the public domain were used then to train machine learning models. And this was the second step. This part of the project was performed by the former PhD student that he already graduated, Norberto Sanchez. Just to briefly summarize what was found, that different machine learning models using extended connectivity fingerprints and RDK fingerprints, and also consensus approaches combining these representations, overall yield to really promising precision and sensibility of the models in terms of classification, binary classification, and target prediction. Now, these models were then implemented in a free web server that is called Epigenetic Target Profiler. This also work of the same former student, Norberto Sanchez. This is a web address. It's totally freely available. And what this server does, it's for an input compound for the user that is interested in just drawing a molecule or getting the smiles. What the server is going to do is basically perform an epigenetic target fishing. This is predict the activity of that small molecule with a panel of 55 epigenetic targets. This is basically an example of the output of epigenetic target profiler. Here we have the input compound as the user entered the compound. Then the server performs an internal standardization of the molecule. And for that compound, display a list of those epigenetic targets for which that compound is going to be predicted as active. Here are links if the user wants to have more information of that particular epigenetic target. And the output also includes a column, the quartile, that is basically kind of a measure, rough measure of the reliability of the prediction. The full details of the implementation are published in this reference. So it was nice that soon after we released the server in the public domain and we put it on LinkedIn. Basically, a few hours later, we got a comment on LinkedIn of Andrei Ursu. He's working at the Scripps Research Institute in Florida, where he or his research group tested the server, and he just a nice comment that the server predict correctly the compound or the compounds that they are working on. So this is the brief story of the first specific aim. Now I'm going to move to the second specific aim, that is virtual screening of compound databases to identify novel inhibitors. We're following, we can say, kind of a standard virtual screening approach. First step is to put together, prepare, curate the compound databases. Second step, the filtering, the in silico selection of the hit candidates, then move these compounds for experimental validation, and then in the fourth step is optimize those compounds. So for the first step, the compound libraries that we have been interested in the last several years are from different sources, including natural products, food chemicals, in-house databases from collaborators, commercial synthetic libraries, and also focused libraries. For the filtering, for the selection, we're using a series of different approaches, similarity searching, molecular docking and dynamics, and more recently, the machine learning models, as I just mentioned, implemented in epigenetic target profiler. To experimentally validate, our first approach is to perform enzymatic inhibition assays, and here we are open to collaborations to further test our compounds in other more complex systems. And for the last part, also through collaborations with medicinal chemistry groups in Mexico and other countries, is basically doing analogs of those compounds and testing the analogs. So I like to spend a few more words about the compound libraries. These are specifically the natural products and food chemical libraries that we have been recently using. Coconut, like Professor Marcus very nicely and other speakers introduces earlier. Biofakim is a database of natural products from Mexico. This database is also freely available, and food database. That is a public collection of roughly 22,000 food chemicals. And we're also exploring food chemicals because we know that epigenetic mechanisms are regulated by food that we're taking every day. Datasets from collaborators are just a couple of examples. It's a collaboration with a group in Mexico that are doing synthetic analogs of caffeic acid, and with a group of Professor Alexander Carnon in Canada. He's also working in small molecules. So we are doing the computational analysis of these sets of molecules. The commercial source, as many other groups, were relying on zinc. And also, kind of recently, several chemical companies have released at least the chemical structures of focused libraries. This is just one example of Otava that has put together a focused library of compounds hitting different epigenetic targets. So on the next slide, I'm going to focus on the virtual screening of focused libraries. And as I mentioned before doing the screening right away, the first step is to curate and characterize the contents of those libraries. So this is a project in where the student Alexis Padilla has been actively participating, and other students as well. The first step here was to collect all the databases. After data curation, there are slightly more than 53,000 molecules. The table here shows the different chemical vendors and the number of compounds per chemical vendor before and after data curation. So as part of the characterization of these epigenetic focused libraries, this picture here shows a visual representation of chemical space. This example is using principal component analysis of 6 properties of pharmaceutical interest, basically Lipinski and Weber's properties that are associated with these rules. In this plot, each focused library is identified with a different color, and the take-home message of this visualization are basically 2. Compound libraries have drug-like properties. Well, this is not really surprising because we know that chemical vendors pay a lot of attention on suggesting drug-like compounds, but anyway, it's always nice to check. And also this analysis suggests that the diversity of those epigenetic focused libraries is different, varies from library to library. Also part of that characterization was visualize the chemical space using other approach. Here we're showing an example of what is called a constellation map. The full details of this methodology, constellation map, the code is freely available. It's published here in this 2019 reference. In this constellation plot, each circle represents not one specific compound, but represents a series of analogs that shares a chemical scaffold. Depending on the library, there will be a series of analogs that are heavily populated, that have many more compounds, and this is just visually represented with data points that are larger. Just for comparison with the terms in cosmology, so we know that in the cosmos there is a polar star, a reference star. So here in this visualization, we are identifying a reference molecule. This is our polar star in the constellation map space. This is basically, structurally speaking, the most representative scaffold of all the libraries that we are comparing in this particular example. Now, in the bottom part of the slide, we are color coding the different specific chemical libraries and identifying what a region of these constellation maps is occupying different libraries. For instance, the library from this vendor, Chemical Diversity, is quite diverse, covers a wide region of the chemical space, and there are other smaller libraries, for instance, this one from Asinex, that covers a specific region of chemical space, but kind of complement the space of chemical diversity. And similar analysis, similar conclusions can be done comparing the constellations maps from different specific libraries. So after we characterized the libraries, the next step was doing the filtering, the screening itself. So this is a brief summary of the workflow that was implemented. The first step was filtering all curated compounds using epigenetic target profiler. But for this end, we use the scripts, of course, not the web server. It's extremely much faster. And from this filtering, we identify around 119 compounds with the best predictions. Next step was to do the consensus docking of this number of molecules with two docking programs, MOE and AutoDoc-BINA, and then the results were rescored using a novel approach that was developed by Norberto Sanchez that is called Extended Connectivity Interaction Features, or ECIF. All the details of ECIF are published in this reference. The table here shows some of the hit compounds that were selected. At the end, were selected 20 compounds for enzymatic inhibition assays. Here in this table, we see the docking scores and the rescoring results from ECIF. Now, moving forward to the experimental validation, at least enzymatic inhibition assays, the first step, we identify several quinolines that have an inhibition range between 10 and 40%. This analysis is done at one single kind of high concentration of the compound, just to have a sense of what molecules, what scaffolds are the most promising. And quite interesting in this analysis, identify a couple of activity cliffs that are highlighted here in different colors. The compound on the left is practically identical to the compound on the right. The difference I'm just highlighting here is two methyl groups. When the two methyl groups are present in the structure, these compounds, it's kind of an inhibitor with 40% of inhibition. But when these methyl groups are replaced, of course, by hydrogen atoms, the compounds becomes not an inhibitor, but an activator of the enzyme. And we are still kind of wondering and working on the modeling, trying to explain why this big change in the activity profile. Perhaps the most striking result from this experimental validation was the identification of this couple of quinazolines. One of them showed that an inhibition of DNMT1 with an IC50 of 30 nanomolar, and the second one, this, as you can see, is quite analog structure, had an IC50 of 81 nanomolar for DNMT1. Both compounds are roughly 10 times more potent than the internal control, S-adenosyl homocysteine, that has an IC50 or 0.34 micromolar. So this level of activity is kind of similar, comparable to the activity of the hit identified from GlaxoSmithKline that I showed previously. Both quinazolines are kind of selected towards DNMT1, at least the inhibition with DNMT3A is much lower, and both of them do not inhibit DNMT3B. Now, in searching in the literature, it was kind of interesting to see that both quinazolines have reported activity with another epigenetic target, an epigenetic reader, G9A, and both of them, here is one example, is also quite potent with an IC50 of 6 nanomolar. So these compounds are dual inhibitors of DNMT1 and also of G9A. In order to start investigating the mechanism of inhibition, Edgar Lopez did docking and molecular dynamics studies. And just to briefly summarize the results, the analysis of these in silico studies that Edgar identified was to propose that the quinazoline is binding to the regulatory domain, the CXX domain. This domain, as you may recall, CXX, well, the regulatory domain is there in 3A and 3B, but the CXX portion is not present in 3A and 3B. Also, experimentally speaking, we did competition assays with the substrate. In this case, is the DNA, and the results show that this specific quinazoline did not compete with DNA with a catalytic site. So putting both results together, seems that these compounds are not binding in the catalytic site, and again, the modeling results strongly suggest that are binding in the regulatory domain. Of course, we need to perform additional studies, but I think we're in a nice direction in this case. So in this project, a couple of perspectives here. It's to perform rationally multi-target epigenetic drug discovery to descend Eli Fernandez, Fernando Prieto, and Michelle Espinosa. We're working together to develop machine learning models to predict the dual inhibition of DNMT1 and HDAC and other epigenetic targets. Other major perspective in this area is the novel design with different DNMTs, and this is a project of Diana Prada. Now, I'm going to move to pretty much the last part of the talk. And very briefly here, I like to give this really personal view of the challenges of chemoinformatics, and this personal, I could say, summary is taken, of course, from many scientific publications. For the sake of discussion, I'm dividing these challenges in 4 major areas: challenges related to chemical space, biological space, methodological challenges, and other challenges associated with human factors. And what I'm going to present here is basically a brief summary of this opinion paper that is published in Frontiers in Drug Discovery. So a few challenges concerning chemical space. I think the scientific community is aware that we need to expand the medicinal relevant chemical space. Part of this expansion is being actually actively going on through the rational design and screening of large and ultra-large chemical libraries. Also, the scientific community is repurposing existing libraries, not only from approved drugs, but also reusing or repurposing in-house collections. Also, as was mentioned just in the previous talk, we know that it's important to revisit our screening campaigns, learning from the results, learning from the advances that is published in the literature, and rescue missing hits from past screening efforts. Also, I think we also need to keep exploring neglected regions of chemical space. There are compounds that are typically not considered for drug discovery programs. For instance, molecules containing metals. We know that those type of compounds are more problematic, but still are promising for some specific targets. Now, a few challenges related to biological space. Also, the scientific community is working more and more in multi-target drug discovery. Also paying attention to dark targets, those targets that are not that popular or frequently explored. Also, there is big concern that is challenging target protein-protein interactions. And again, the scientific community, I think we need to keep investigating those targets associated with rare and neglected diseases. There are several methodological challenges. Perhaps I'm not going to just read all of them just for the sake of timing, but just to mention, emphasize the last challenge here. Basically, especially now that we are in the big era and a lot of artificial intelligence going on, data curation and ensuring data quality is fundamental. Additional challenges with this chemical space, biological space, and methodological challenges is human factor. Just want to highlight a couple of them. Inside research organization, research groups, it's quite fundamental to improve our communication between our research groups. Other challenge is to be responsible and ensure rigorous dissemination, transparency, reproducibility of the data that is put out there, and education and training of individuals and research teams is also a must. Perhaps I could say kind of the grand, in quotes, a challenge in this era of artificial intelligence is use of any computational methods in a rational way to avoid these hypes. And I kind of borrowed this phrase from a different field, but I think really applies to what is going on right now, that unrealistic expectations sometimes are quick roads to disappointment. So the challenge here is to keep our expectations realistic. Just brief summary. We see that we're convinced that chemoinformatics is an independent discipline itself. In this talk, we talk briefly about epigenetic drug discovery, the development of epigenetic target profiler, and the identification of small molecules targeting DNMT1. I could summarize, again, personal view of the chemoinformatics challenges of chemoinformatics at CAP is revisit and expand the chemical and biological spaces, ensure data quality, because this is going to affect several methods that we use. And also we need to keep in mind to keep effective our communication and keep working on education and training. So with that, I would like to finish, and on behalf of all of our research team, the FACIM, want to thank the collaborators that are working on the assays, on the synthesis. Also many thanks to the funding institutions. And probably going to take half a minute now to invite all of you, if you are interested, in Mexico at UNAM, we are organizing an online free event. It's a colloquium focused on chemoinformatics and artificial intelligence. There are speakers from different environments, saying academia, from big pharma, small pharma, and other research non-for-profit institutions that will be talking about progress and also challenges of chemoinformatics and AI to develop peptides, natural products, and drug candidates. With this, I'm going to stop. I want to thank you again for your attention and again for your invitation to participate in this wonderful symposium. Thank you.
Thank you very much, Professor Medina. Now it's time for questions. Those can be asked either in the chat or directly, please. Very nice talk. In your opinion, what is the main challenge for screening large databases?
Well, thank you. So I would say the first challenge is to curate those databases. So ensure that the databases have the correct chemical structures. All these curation protocols are really well implemented. Like doctoverism, protonation states, and so on and so forth. And also, like, keep in mind that perhaps not a single method is going to give the ultimate hits. Like, to think about a combination of different methods, of course, depending on the information that is available to suggest hits compounds.
Thank you very much. Any additional questions?
Yeah, may I ask?
By all means.
Yeah. So, Jose, I just read this paper in the Drug Discovery today. Yes, sir. Yeah.
Oh, okay.
Nice. The structure and activity relationships in drug discovery. It's very good, insightful paper. But the question actually is very old, because I remember when we have some communication in the former QSAR society, it was the same question, and Hugo Kubinyi commented that please look in the Journal of Medicinal Chemistry. You see a lot of tables. Some compounds are active, some other are inactive. So there are many information about inactive compounds, but probably they are filtered out when it's annotated by Campbell or some other integrated sources. Is it true?
Yes, exactly. Well, thank you so much for the comment. I would like to mention that that idea to putting together that paper actually came from a PhD student, Edgar Lopez, that I think is here in the audience. So the credit goes for him. And yeah, basically the idea is to emphasize that in large public databases, there is few information concerning inactive molecules.
Yeah.
Basically remind the community to disclose inactive molecules.
Thank you.
Thank you.
Okay, I will have one last question for the sake of time. So in your very nice section of DNT1 inhibition, so I understand that your compound 30 nanomolar came out of your silica screen, but there was a sort of competitive publication in JMed Chem. So how those people came across this chemotype?
So it was from, they were working on kinazolines for other projects and then start targeting those compounds with a panel of epigenetic targets and identify that were potent inhibitors with gene A. And so it's kind of rational and also, and then keep developing the compounds. That group now has published several papers refining those structures, doing a lot of SAR.
I see. But in your study, it came out of natural products library or synthetic library? What's the origin of this model?
Focused libraries for comment from a commercial vendor.
Okay, so it's possible that probably those libraries been constructed on some prior knowledge of this class of compounds inhibiting something. So did you have a chance
exactly.
to trace it down to why these compounds, because they seem fairly developed, like a lot of substitutions, a lot of rings. So it looks like they've been rationally approached before. So I was wondering what was done.
Yeah, that's a good point. Yeah, I'm as you mentioned, I agree with you. Probably this chemical vendor just kind of spot those compounds in the literature and now are actually making those compounds available for experimental screening.
I understand.